Welcome all to AK Bio Lecture Series. In the last few classes, we discussed the cell cycle and regulation. Today's topic is on the internal cell cycle regulation by the checkpoints or checkpoint proteins. Before discussing the core topic, a flashback into the history about the discovery of checkpoint proteins might be an interesting thing. Madame Louise Bahr, a Belgian neurologist who published a case report in 1941 describing a 9-year-old boy with progressive cerebellar ataxia and cutaneous telangiectasia. Ataxia is a neurological condition, neurological sign consisting of lack of voluntary coordination of muscle movements. And telangiectasia is a condition in which widened venules cause thread-like red lines of patterns on the skin. For the next couple of decades, the clinical condition with the combination of ataxia, both ataxia and telangiectasia, was referred to worldwide as Louise Bahr syndrome until 1964. Then, another scientist, Boder and Sedgwick, described 8 patients with classical symptoms of ataxia and telangiectasia and suggesting the name ataxia telangiectasia. Ataxia telangiectasia, in short form, it is called AT, is an inherited recessive disorder characterized by a host of diverse symptoms, including a greatly increased risk for certain types of cancer. In 1988, Gatti and his co-workers mapped the ataxia telangiectasia gene on the Q arm of chromosome number 11. In 1995, another scientist identified the defective gene responsible for AT, that is ATM gene. During the late 1960s, following the deaths of several individuals undergoing radiation therapy, it was discovered that patients with ataxia and telangiectasia symptoms are extremely sensitive to ionizing radiations. So, what might be the role of ATM in the cell? So, ATM, this is the ATM gene protein, and ATM gene encodes a protein. This is the protein. It is a protein kinase, that is a serine threonine kinase, protein kinase, that is recruited and activated by DNA double-strand breaks. So, this protein will be activated only in the condition of DNA double-strand breaks. Note that it is double-strand breaks the ATM protein can sense. The presence of a single double-strand break in one of the cell's DNA molecule is sufficient to cause rapid large-scale activation of ATM molecule, causing the cell cycle arrest. So, what a sensitive and sensible protein it is. Then, how a normal cell will respond to the radiation? In an experimental condition, a researcher irradiates a normal cell during the G1 phase of the cell cycle, and it delays the progression into S phase. So, similarly, cells irradiated in S phase delay further DNA synthesis, whereas cells irradiated in G2 phase of the cell cycle delay its entry into mitosis. So, what is the inference of these test experiments? From the observation of the experimental condition in yeast cells, scientists proposed the concept that the cells possess a checkpoint as part of their cell cycle. What might be the job of checkpoints in the cell. A checkpoint is one of the several points in the eukaryotic cell cycle at which the progression of a cell through its cell cycle is regulated at distinct stages until conditions are favorable. Checkpoints are surveillance mechanisms that halt the progression of the cell cycle in emergency conditions. So, the conditions could be either the damage of the chromosomal DNA or damage in the certain critical processes such as DNA replication during S phase or chromosome alignment during M phase. Checkpoints ensure that each of the various events that make up the cell cycle occurs accurately and in the proper order. Many of the proteins of the checkpoint machinery have no role in normal cell cycle events and are only called into action when an abnormality appears. When any abnormality is sensed by sensor proteins of the cell, then it triggers a response that temporarily arrests further cell cycle progress. The cell can then use this time delay to repair the damage or correct the defect rather than continuing to the next stage. If the DNA is damaged beyond repair, then what will be the option in front of this cell? In that situation, the checkpoint mechanism will transmit the signal that either to the apoptosis or senescence of that cell. There are internal control mechanisms that operate at three main cell cycle checkpoints. The first checkpoint is near to the end of G1 phase of the cell cycle, that is G1 to S phase transition stage. And the second checkpoint is at the G2 to M transition stage. And the third one is during metaphase stage. So these are the three checkpoints. You can see here the three checkpoints, isn't it? 1, 2, 3. How does a cell stops its progress from one stage of the cell cycle to the next? To answer this question, we will briefly examine two well-studied pathways available to mammalian cells to arrest their cell cycle in response to DNA damage. The cell chooses the pathway depending upon the kind of damage DNA hold. The damage might be either single strand break or double strand break. The first pathway is also called ATR-mediated pathway. The cell opts for this pathway if the aberration is single strand break of DNA. And second pathway is ATM-mediated protein-mediated pathway, and the cell enters or the cell opts for this pathway if the aberration is double strand break of the DNA. Keep in mind that all these pathways are multi-step processes and they all are part of extraordinarily complex intermediate or interconnected signaling networks. Let's discuss the first pathway, that is ATR-mediated pathway in detail. Consider a cell preparing to enter mitosis is subjected to UV radiation, and means now the cell which is in G2 to M phase transition stage exposed to UV radiation. And what would be the result of the exposure to the UV radiation on that cell? Of course, there will be a chance for the aberration in the DNA. Most probably, the chance for single strand breaks is there. So, single-strand DNA is a DNA structure that is commonly induced by DNA damage and by interference with the DNA replication. In addition, single-stranded DNA gaps are induced by several types of DNA repair mechanism such as nucleotide excision repair, then mismatch repair and long patch base excision repair. Single-stranded DNA is also present at double-stranded breaks that have been trimmed by exo or endonuclease through a process called resection. Here we are not discussing how the single-stranded DNA is formed during repairing and all. I hope you already learned those events when you studied the DNA replication and repairing mechanism. In this context, you just understand and remember one thing, that the single-stranded DNA formation is not only related with the UV exposure, but it will also be resulted from the replication stress or as part of DNA repairing mechanisms. You can see here the conditions that leads to single-stranded DNA formation. Here, whatever might be the situation, if the cell is in G2 to M transition phase, the cell will handle the situation in same method. As you know, in normal cells, cyclin D, CDK4 or 6 complex are essential for entry into G1 phase, and cyclin A CDK2 complex is critical in the S phase, and cyclin A CDK1 complex promotes G2 to M transition, and in the late G2 phase, cyclin B replaces cyclin A, and cyclin B CDK1 complex triggers mitosis. So when the cell senses some kind of DNA aberrations during the G2 to M phase, then the cell must turn off or inactivate the cyclins and CDKs at this point that promote G2 to M phase transition, and the cell should keep in cyclin A CDK1 complex in inactive state. So these cyclins and CDK will be inactivated, keep in inactivated state for the arresting of the cell cycle. Then only the repairing system can take the control of the cell. How the cell achieves the inactivation process? First, just after the event of DNA damage, a series of protein come into the scene in a sequential way. Before starting the discussion in detail, remember two things. First one is, now the cell is in G2 to M transition phase. So now the cell is with a clear nuclear envelope, clear nuclear membrane. You can see here the picture of G2 to M phase cell. Here you can see the clear nuclear membrane. The second point to remember is about the location where the events are taking place or happening. The first part of the event occurs inside the nucleus. Actually, it occurs inside the nucleus. Then after that, some events will occur in cytoplasm. Then the final part of the process will proceed inside the nucleus. So the process will start from the nucleus, then to cytoplasm, then again it will come back to the nucleus. So first location is inside the nucleus. Which are the events takes place inside the nucleus when a cell exposed to DNA damage in G2 to M phase of the transition phase? We are going to answer to this question. In all eukaryotes, single-stranded DNA that is induced by DNA damage is first detected by a replication protein A, that is RPA, RPA is the full form of RPA is replication protein A, which is a single-stranded DNA binding protein complex. You can see here the recruitment of RPA protein to single-stranded DNA strand, single-stranded DNA region. Here, in the next step, you can see the attachment of RPA on the single-stranded DNA. You can see here exactly at this point. The next step, ATR interacting protein. Then here you can see here the ATR interacting protein, that is ATRIP, which is the regulatory partner of ATR, and it binds directly to these two proteins, that is ATRIP and ATR, directly binds to RPA coated single-stranded DNA and thereby enables the ATR interacting protein complex to localize to the sites of DNA damage. You can see here the attachment of ATR-IP complex on the RPA protein. The localization of ATR and ATR-IP to sites of DNA damage is not sufficient to activate the DNA damage checkpoint response fully. Instead, it requires the function of additional ATR regulators, including the RAD17, RAD9, RAD1 and HUS1. The RAD17, RAD9, RAD1 and HUS1 complex is also called 911 complex. Following the recruitment of ATR, ATR-IP and the 911 complex, the kinase activity of ATR is stimulated. As a result, the kinase activity of ATR is stimulated. Next, ATR protein phosphorylates and activates a checkpoint kinase called CHK1, and phosphorylated CHK1 activated CHK1 phosphorylates CDC25 on a particular serine residue inside the nucleus. In the last class, you studied that CDC25 is a phosphatase, and the active form of CDC25 protein will remove the inhibitory phosphate at tyrosine 14 and 15 of CDK1 at the end of G2 phase and triggers the initiation of mitosis. So, active form of CDC25 is a mitosis promoter. This active form of CDC25 is the mitosis promoter. So, the cell must inactivate this CDC25 to block the progression into the M phase from G2 phase. When CHK1 phosphorylates CDC25, on a particular serine residue inside the nucleus, CDC25 become inactive. So, after the phosphorylation event of CDC25 will be inactive. The phosphorylated CDC25 or the inactive form of CDC25 will now transported out of nucleus into the cytoplasm, and the phosphorylated CDC25 is bound by an adapter protein here. You can see here the adapter protein, isn't it? In the cytoplasm. In adapter bounded form, in the adapter bounded form, CDC25 cannot be transported back into the nucleus again. So, at this moment, what is the situation inside the nucleus? There is no CDC25. Here, there is no CDC25. Now, the CDC25 is inside the cytoplasm as bounded with the adapter protein. So, there is no CDC25 in the nucleus, to activate CDK1. What is the function of active form of CDC25? It will activate CDK1. So in the absence of CDC25, CDK1 will not get activated. As a result, the cell will not progress into M phase. So this is how ATR kinase is activated and the cell arrest in G2 phase of the cell cycle. Actually, in this class, I am not explaining the mechanism of CDC25 transport across the nuclear membrane. This is all about ATR-mediated pathway. Next is ATM-mediated pathway. ATM-mediated pathway. Consider if a cell is in G1 phase. This is the cell which is in G1 phase and it is exposed to the ionizing radiation. Actually, the radiation exposure resulted into the double-stranded break of DNA in that cell. Here, a protein complex called mRNA senses the double-stranded break in that cell, and mRNA, here the mRNA recruits and activates ATM protein. Here you can see the ATM protein, and activated ATM phosphorylates and activates another protein, checkpoint protein called CHK2. And activated CHK2, this activated CHK2 in turn phosphorylates, this activated CHK2 in turn phosphorylates the transcription factor called P53. And this is the P53 protein, and this is the three-dimensional structure of P53, and a protein, this protein which is considered as the guardian of the cell because its role in the checkpoint. So this is very crucial protein in the regulation of cell cycle. Actually, 50% of all human tumors show evidence of mutation in the gene that encodes the P53, this P53 protein, which reflects the importance of the control of the cell cycle, of this protein in the control of the cell cycle. When the P53 protein is activated in the cell, when this P53 protein is activated in the cell, it functions as a transcription factor of different genes that regulate the cell cycle. And one of the important genes that regulated by the P53 protein is this P21 gene. And in the last class, when we discussed, this is the P21 gene, you can see here the P21 gene. In the last class, we have discussed the cell cycle regulation by CDK inhibitors, and we categorized the inhibitors into two. Which are they? ING4 family inhibitors and CYP-KIP family inhibitors. Actually, I mentioned in the previous class that the P21, P27 and P57 are the CYP-KIP family of proteins form heterotrimeric complexes with cyclin D, cyclin E and cyclin A dependent kinase complexes. For more details, watch previous class video. Here in G1 to S checkpoint, one of the inhibitor of CYP-KIP family, that is P21 gene, is activated by P53 transcription factor, which leads to the transcription and translation of P21 gene and subsequent inhibition of CDK2 cyclin E complex. Actually, in the diagram you can see here the P21 protein inhibiting the CDK2 cyclin E in the G1, in the late G1 phase, that is exactly at checkpoint of G1 to S transition phase. And this is all about the checkpoint mediated cell cycle regulation, and hope you like today's class, and thank you very much.